Hello, 7th standard. Have a good day. Now we will continue video session 10. And it is a continuation of circulatory system. Previous class we have learnt about structure of human heart, then functioning of human heart. In structure of human heart we have learnt as external structure. External structure enna sollணும், one's own fist size வந்து one's own fist. It is made up of cardiac muscle. Cardiac muscle வந்து myogenic in nature that initiates rhythmic activity of the heartbeat. Then heart is covered by two-layered pericardium which is filled with pericardial fluid and it protects and acts as a shock absorbers. And then heart itself supplied with coronary artery and coronary vein with the blood circulation. I hope you remember this. Then valves internal structure as chambers of the heart, right atrium, left atrium, right ventricle, left ventricle. Next, valves of the heart, tricuspid valve, right atrium opens into right ventricle, left atrium opens to bicuspid valve, left atrium opens into left ventricle. Bicuspid valve is also known as mitral valve. There is another one, semilunar valve, through which from the left ventricle oxygenated blood is sent through the aorta distributing to various parts of the body. I hope you remember this. And blood vessels, they are aorta supplying blood to various parts of the body. Vena cava, superior and inferior, collecting impure blood from various parts of the body. I hope you remember this. These are blood vessels and these aorta branching into arteries which in turn arteriole. Same way vena cava branching into veins which in turn ending in venules, capillaries. Sorry? And then functioning of heart. Now here in the heart, what is circulated? It is the blood. Blood is a liquid connective tissue circulated within the body and it carries all the transporting substances as food with the nutrients and waste materials and then gases for exchange and then hormones and anything to be circulated, to be transported, then it is the blood. I hope you are following. Yeah? So now human blood is a liquid tissue and it is divided into two regions, two categories. One is fluid plasma and then it is solid corpuscles. Fluid plasma and solid corpuscles. Okay. Now this fluid plasma, it is fluid in nature, semi-fluid, solid and liquid and it is a semi-fluid. And then these are the contents of blood plasma. These are the contents of blood plasma. What do we know? First, water. Anything to be circulated now, it is with the water. Water may be in different form as blood the corpuscles of fluid plasma. Water is present. Water is as a solvent and suspending medium for blood components. Water is a solvent and suspending medium for blood components. And then it has plasma protein as albumin, globulin and fibrinogen. Plasma proteins are albumin, globulin and fibrinogen. Where albumin, it maintains osmotic pressure and globulin, it is meant for antibody formation and fibrinogen, it is taking part in blood clotting. So the plasma protein edalana albumin maintaining osmotic pressure, globulin antibody formation, fibrinogen is blood clotting. This is for plasma proteins. And then these ions as sodium, potassium, calcium, magnesium and chloride ion, they are for osmosis and acid base balance. Edala sodium, potassium, calcium, magnesium and chloride ions. They are meant for osmosis and acid base balance. Whereas iron, phosphate, hydrogen ion and bicarbonate ions, they are acting as a buffer. Then nutrients present in the blood plasma, they are glucose, amino acids, triglycerides, cholesterol and vitamins. Nutrients, they are glucose, amino acids, triglycerides, cholesterol and vitamins. Glucose, they are for carbohydrates, amino acids for protein, and triglyceride and cholesterol, they are for fatty substances and vitamins. Sure? Then waste products, urea and uric acid, they are excreted through the kidneys, excreted by the kidneys. And creatinine, ammonia, bilirubin, they are the creatinine and ammonia. Sorry, urea, uric acid, creatinine and ammonia, they are excreted by the kidneys. Urea, uric acid, creatinine and ammonia. Then bilirubin, it is the breakdown product of erythrocyte. And lactic acid, it is formed due to anaerobic respiration. It is accumulated in the muscle cells due to anaerobic respiration. Clear? Then gases are exchanged. Oxygen for aerobic respiration, carbon dioxide, it is as waste product of respiration, and nitrogen, it is a inert gas. And then regulating substances, they are hormones and enzymes. Enzymes are digestive enzymes, where the food taken in, they are digested. And hormones are biochemical substances which are essentially needed for all the metabolic activities. I hope you understand. So this is for the plasma, content of the plasma and the functions. Are you clear? Then solid corpuscles, they are as RBC or erythrocytes. RBC or erythrocytes. This is leukocytes and karyocytes. Leukocytes and megakaryocytes. Okay, so they are the blood platelets. Leukocytes and karyocytes. They are as blood platelets. I hope you understand. So solid corpuscles, they are RBC, red blood corpuscles, or erythrocytes. WBC, white blood corpuscles, or leukocytes and blood platelets. I hope you understand. So now this features of this RBC and WBC, size and shape, it is disc shape biconcave. It is disc shape and biconcave. And then it is lifespan 100 to 120 days. Lifespan of RBC 100 to 120 days. and it is produced by bone marrow. It is produced by bone marrow. And then it is having the red pigment hemoglobin. Having a red pigment hemoglobin. I hope you are following. So, size and shape of erythrocyte, it is solid, disc shape and biconcave. Its lifespan is 100 to 120 days. It is produced by bone marrow and destroyed in liver and spleen. And it is destroyed in liver and spleen. What does it mean? After 100 to 120 days, the RBCs will be destroyed and it is reaching the liver and spleen. In the liver, destroyed RBC, the bilirubin and biliverdin, the breakdown product of RBC or the hemoglobin. And then it contains red pigment hemoglobin. I hope you follow this. And then in mature mammalian RBC, there is no nucleus. In matured mammalian RBC, the nucleus will not be present because it itself is carrying the oxygen. I hope you understand. Whereas in leukocyte, it is irregular. The size and shape, it is irregular, and then its lifespan is 2 to 3 weeks, and it is also produced by bone marrow, and it is destroyed as pus because it is an organ of defense. It is a type of cells meant for defense, and then it is having the nucleus. On WBC, it is having the nucleus. Okay. Now this WBC leukocyte, it is of two types. One is granulocytes and another one is agranulocytes. Granulocytes and agranulocytes. Granulocytes are neutrophils. Three types: neutrophils, basophil, and eosinophil. Neutrophil, basophil and eosinophils. Okay. Whereas agranulocytes, they are lymphocytes and monocytes. Lymphocytes and monocytes. And they are irregular in shape like this. In the monocyte. And they are provided with the nucleus. They are provided with the nucleus like this. They are with the nucleus. And this is called lobed nucleus. Nucleus of WBC, it is known as lobed nucleus. I hope you understand this. So WBC, also known as leukocytes, they are irregular in shape, 2 to 3 weeks lifespan, produced by bone marrow, and there is no hemoglobin pigment present, and it is of two types as granulocytes and agranulocyte. Granulocytes are neutrophil, basophil and eosinophil, and agranulocytes are lymphocytes and monocytes, and they take part in defense, fighting force. They are as police force. Okay, and then it is having lobed nucleus. That is RBC nucleus, mammalian RBC nucleus கிடையாது. Mature RBC nucleus கிடையாது. Whereas leukocytes, it is having prominent nucleus, lobed in a dividend. So it is called lobed nucleus. I hope you understand. So then platelets, it is also called megakaryocytes. Megakaryocyte, and it is also irregular in shape, and it is meant for blood clotting. Presence of platelets with the fibrinogen of plasma protein, they take part in blood clotting. What is blood clot? When any part of the body is injured, the blood is shed, and that blood should be stopped, bleeding should be stopped immediately. That is called blood clot. Otherwise, person will be suffering from a disease called hemophilia. I hope you understand this. So this is forming the composition of blood or structure of blood. It is a definite 10 marks, or it may be as 5 mark. Plasma content, describe the liquid portion of the fluid part of the blood. You have to describe about plasma. If you say describe the solid corpuscles of blood, you have to say about RBC, WBC and blood platelets. I hope you understand. Plasma blood plasma, water, it is acting as a solvent and suspended medium for blood components. Then it has plasma protein, albumin for osmotic pressure, globulin, antibody formation, fibrinogen, blood clotting. Then ions as sodium, potassium, calcium, magnesium and chloride, they are for osmosis and acid balance. And then iron, phosphate, hydrogen and bicarbonate ion, they are for buffer. Then we have learnt about nutrients, waste products, gases, and then hormones and enzymes. These are for blood plasma. And then RBC, WBC and blood platelets. I hope you are following. It is the continuation of the blood circulation, the shorter duration, the complete. We will continue in the next video session. Thank you. Hello, 7th standard. We continue transportation in animal, video 10. Yesterday we have been learning previously in video 10 composition of blood and other continuation. Okay, what have we learned? Composition of blood. We have learned as plasma, blood plasma, fluid portion, and then solid corpuscles as RBC, WBC, and blood platelets. In that RBC, it is also known as erythrocytes, and WBC, it is also known as leukocytes, and blood platelets, it is also known as thrombocytes. Blood platelets, it is also known as platelets. It is also known as thrombocytes. Okay, and then we have studied size and shape. Then produced by bone marrow. Lifespan is 100 to 120 days in RBC, 2 to 3 weeks in WBC and thrombocytes. And then RBC takes part in respiration, where it is containing the red pigment hemoglobin. I hope you remember this. This red pigment hemoglobin combines with oxygen to form oxyhemoglobin. I hope you understand this. So as a result, in mammalian RBC, it lacks nucleus, very important. Whereas WBC, it is having lobed nucleus. Lobed nucleus. Blue color, right? And then this blood platelets, it It is also known as thrombocytes. The thrombocytes they take part in blood clotting. I hope you understand. So when you learn this video number 10, the picture for RBC, WBC and blood platelets will be available that you can understand. And in the WBC, the blue, the lobed around it, you have to label it as lobed nucleus. I hope you understand. And then you should know about stethoscope and sphygmomanometer. Stethoscope, normally it is for heartbeat. It is an instrument to hear the heartbeat sounds, and it is due to the inhalation and exhalation of air in the lungs and the respiratory pathways. I repeat, stethoscope is used to hear the heartbeat sound due to inhalation and exhalation of air in the lungs and also air pathways. I hope you understand. And it also indicate the movement of the stomach. And actually, it is very useful diagnostic tool to help localize the problem. In a breathing line problem, the doctor check the stethoscope, right? It is also used along with sphygmomanometer. Blood pressure பார்க்கிறது and the sphygmomanometer இதையும் பார்ப்போம். சரியா? ஸோ நம்ம புக்கில் ஒரு ஆக்டிவிட்டி இருக்கு. அதாவது யூ டேக் எ ஃபனல், அந்த ஃபனலோட ஒரு ரப்பர் டியூப் சொல்லணும், ஒரு நீல ரப்பர் டியூப் சொல்லணும். சொருகிட்டு இந்த ரப்பர் டியூப் மூலம் நீ ஏர் கொடுத்து இந்த ஃபனலை இங்கே அந்த இடத்துலலாம் வச்சு பார்த்தேன்னாக்க யூ குட் ஃபீல் த இன்ஹலேஷன் அண்ட் எக்ஸ்ஹலேஷன். தட் இஸ் இம்ப்ரவைஸ்ட் ஆக்டிவிட்டி ஃபார் stethoscope, instrument stethoscope. சரியா? And this alt integration நமக்கு ஒரு சாப்டர் உண்டு. நம்ம ஒரு டாபிக் இருக்கு. ஆக்சுவலா நம்ம for every lesson we have to do activity, enrichment activity. So accordingly you have to prepare. நம்ம போர்ஷன் நிறைய முடிச்சிருக்கோம். நான் இதெல்லாம் சொல்லியிருக்கேன் உனக்கு சரி. டைஜஸ்டிவ் சிஸ்டம் மாடல் ரெடி பண்ணேன். ஹியூமன் ஹார்ட் model you please get ready, and excretory system adande working model pannanam. Same way this stethoscope you have to get ready. Enalla human digestive system, second one human heart, third one excretory system. In the model you have to prepare, get ready. So that is for enrichment activity. Now we will see uses of stethoscope. Stethoscope helps to find the normal lub versus abnormal heart sounds. That is called heart murmuring. That is known as heart murmuring. And also to diagnose valve function. Heart valve function இதை காமிச்சு கொடுக்கும். இதில் QB வரும், அதை புரிஞ்சுக்கும். Right? The stethoscope can indicate fluid in the lungs in case of pneumonia and pulmonary edema. And it can diagnose airway diseases like bronchitis and pleuritis. Bronchitis na inflammation of the bronchi. Bronchi நம்ம படிச்சிருக்கோம்ல, respiratory systemல as trachea branching into two as bronchi. I hope you understand. And then pleuritis. Pleuritis inflammation of pleura. That is pleuritis. Then third one, stethoscope are also used to compare the movements in the normal versus overactive or underactive intestinal tract. Stethoscope are also used to compare movements in the normal versus overactive or underactive intestinal tract. Intestinal tract is small intestine and large intestine of the digestive system. I hope you understand. The next one is sphygmomanometer. This stethoscope normally உனக்கு காமிச்சிருக்கேன், இருந்தாலும் உனக்கு படம் காட்டுவேன். This is stethoscope. Doctors வச்சிருப்பாங்க அது. Right? The next one, it is sphygmomanometer. Sphygmomanometer இந்த இருக்கு. Doctors blood pressure பண்ண பார்க்கிறதுக்கு வச்சிருப்பாங்க. That is sphygmomanometer. It is an instrument used to measure blood pressure. Okay, and during ventricular systole, another arterial blood pressure is the force of pressure. Arterial walls are thick, enna adhu blood flow jaasti irukum. So pressure is the force of pressure which blood is exerting on the walls of the blood vessel. The blood pressure in the artery varies during cardiac cycle. சரியா? So the cardiac cycle is defined as cycle of events that take place during one systole and one diastole of the heart. Again, contraction and relaxation. Systole refers to contraction and diastole refers to relaxation. Systole refers to contraction, diastole refers to relaxation. சரியா? So adula vandu and the force idu varumbodhu that shows systolic pressure and diastolic pressure. I hope you understand this. So uses of sphygmomanometer helps to estimate the state of blood circulation and working of heart. Then sphygmomanometer helps to diagnose pathological conditions such as hypertension and hypotension. This is called sphygmomanometer. I hope you understand. The next transportation is about excretory system. Next transportation is excretory system. What do we know? Excretory system. It is elimination of waste. Normally, this elimination of waste from the body. First one we refer to sweat. Sweat gland secrete sweat. அதுவும் ஒரு எக்ஸ்கிரீட்டரி ப்ராடக்ட். எனக்கு தெரியாம வேகணும் மேல பார்த்தா உப்பா இருக்கும் இல்லையா, அதுவும் ஒரு எக்ஸ்கிரீட்டரி ப்ராடக்ட். அண்ட் தென் செகண்ட் ஒன் கார்பன் டை ஆக்சைடு டியூரிங் ரெஸ்பிரேஷன். Carbon dioxide during respiration. That is also during respiration, oxygen is taken in and carbon dioxide is given out. So that is also a excretory product. And then the third most important is the nitrogenous waste. Nitrogenous waste as urea, uric acid, creatinine and ammonia. Urea, uric acid, creatinine and ammonia, these are all nitrogenous waste products. I hope you are able to follow this. So now the structure of kidney, normally it is a bean-shaped structure. It is a bean-shaped structure. Now this kidney, it is a bean-shaped structure having outer convex and inner concave portion, right? And then outer layer is thick in nature and inner layer, outer layer is thin in nature and inner layer is thick in nature. And then from the kidney is emerging out as a tube called ureter. From the kidney is emerging out as a tube called a ureter. This is ureter, and it is opening into a muscular elastic bag that is called urinary bladder. That opens into muscular called urinary bladder. Okay, and then it is opening outside through urethra. This is kidney. This is excretory system. It is also known as urinary system. Excretory system is also known as urinary system. Okay, so that kidney, it is having excretory system, also known as urinary system, is having a pair of kidney. From there arises emerging out as a ureter. This ureter opens into the muscular elastic bag, that is called urinary bladder, opens outside through urethra. And then outer portion, the outer wall of the kidney, it is thin in nature and the inner is thick in nature. I hope you understand. And then it is normally bean-shaped structure. And the right kidney is slightly lower than the left kidney due to the presence of liver in the position. I repeat, right kidney is slightly lower than the left kidney because of the position of the liver. That is for the excretory system. What do you know for the excretory system? It is as excretory system. It is eliminating what are the products? Nitrogenous products as urea, uric acid and creatinine and ammonia. I hope I am clear. So now the internal structure of kidney, as we know, internal structure of kidney, as we know, as one minute listen, I'll just draw on the board before that. Now this from the kidney is arising blood vessels. From the kidney arising blood vessels like this. From the kidney blood vessel as what I said, renal artery and renal vein. We use the term renal wherever we come across kidney. So we say renal vein, renal artery. Okay. Same way, when it is concerned with the liver, it is called hepatic. We say hepatic artery, hepatic vein. Okay. So now these are the blood vessels supplied to the kidneys as renal artery and renal vein. And above the kidney is present adrenal gland. Adrenal gland enna sollirukken? It is the endocrine gland. Adrenal gland is the endocrine gland. I hope you understand. So now I just draw the diagram and picture idu varum video la. Just understand. Kidney, it is having outer portion and this is inner portion. Now this outer portion, it is as renal cortex and inner medulla. The kidney is formed of two layers, outer cortex and inner medulla. And then this cortex, it is normally somewhat fibrous capsule. It is covered by a fibrous capsule. Right? And then from the inner part of the kidney, it is arising cone-shaped projection. 1 minute. From the inner part is arising cone-shaped projection like this. And this will be arranged like this. The structure will be arranged like this. Okay. So this is called renal pyramid. Pyramid mari shape irukilla. So this is called renal pyramid. Okay. So this portion, renal pyramid, in the portion irukilla. And then in between this renal pyramid is having numerous hand-like structure that is called renal papilla. this is provided with numerous hair-like structure that is known as renal papilla. I hope you understand. So then this pyramidal renal papilla, they converge here, and that is called renal pelvis region. That is known as renal pelvis region. I hope you are able to follow this. So I repeat, internal structure of kidney, it is as outer cortex. This is outer cortex and inner medulla. Outer cortex and inner medulla. And internally it goes like this. Yeah. So this one, it is projecting out like this. So this portion, it is as renal pelvis. And this forming structure like this, pyramid. So this is renal pyramid. And then it gives rise to numerous renal hair-like structures that is called renal papillae. I hope you understand. So this outer cortex, inner medulla, it is formed of numerous functional unit that is called nephrons. So nephrons are the structural and functional unit of excretory system. I repeat, nephrons are the structural and functional unit of excretory system. Are you clear? Then this nephron has, 1 minute, this nephron, it is formed of, now we are going to learn structure of nephron. Now this nephron is having a cup-shaped structure. A cup-shaped structure called Bowman's capsule. Cup-shaped structure called Bowman's capsule. I hope you understand. Now this Bowman's capsule, it is formed of blood capillaries. Bowman's capsule, it is formed of blood capillaries. And that blood capillaries, it is called glomerulus. Bowman capsule is formed of blood capillaries, which are called glomerulus. Otherwise, I say glomerulus is nothing but blood capillaries. Right? Now, these blood capillaries, they are as afferent arteriole. They are as afferent arteriole and efferent arteriole. Afferent and efferent arteriole. What do we know? Afferent arteriole, from various parts of the body, blood collects nitrogenous waste entering into nephron. Okay. Same way, from the after eliminating the nitrogenous waste, it is the elimination is sent out. That is as efferent arterioles. I hope you understand. And then this Bowman capsule and glomerulus together called Malpighian capsule. The glomerulus and Bowman capsule together called Malpighian capsule. And then it is continued as highly coiled tubular structure. It is continued as highly coiled tubular structure, and that is known as convoluted tubule. Tubular structure known as convoluted tubule. So this is called proximal convoluted tubule. Convolution of proximal convoluted tubule, and this is distal convoluted tubule. Distal convoluted tubule. Okay, and then this U-shaped structure without convolution, it is called Henry's loop. I'll continue. So, this proximal convoluted tubule immediately arising from Bowman's capsule. So, that is proximal convoluted tubule that is continued as U-shaped Henry's loop. Here there is no convolution, and you will just see a U-shaped convolution. Okay, and then further again convoluted tubule, that is called distal convoluted tubule. Tubule na kocha tubular structure, right? When it is towards Bowman capsule, I will get a proximal end. So it is called proximal convoluted tubule. When the tubule is again convoluted away from the Bowman capsule, it is known as distal convoluted tubule. I hope you understand. So this is the structure of nephron. As I said, kidney is formed of outer cortex and inner medulla solitarium. So this is the fibrous capsule. Outer cortex, it is lined by fibrous capsule. Now this cortex portion, Bowman capsule will be present. In the renal cortex portion of the kidney, Bowman capsule will be present. And continuation of that, proximal convoluted tubule, Henry's loop and distal convoluted tubule will be present in the medulla region. I hope you understand. So this is forming the structure of nephron. And this nephron is considered as structural and functional unit of excretion. And this nephron only is excreting or eliminating the urine. Okay, so in human, daily around 170 to 180 liters of urine is excreted. Just imagine 170 to 180 liters of urine is eliminated. At the same time, during urine formation, normally we say it is mechanism of urine formation. For that, it takes 3 steps. Mechanism of urine formation, it takes 3 steps. One is glomerular filtration. One is glomerular filtration. This is glomerular filtration. Second one is tubular secretion. Tubular secretion. And third one, tubular excretion. Otherwise, tubular reabsorption of chalalam. So in that 170 liters of urine excreted every day, only 1.5 liters of urine alone is sent out. All the remaining useful substances, they are reabsorbed. As in when the urine is formed, glucose, amino acids and different ions as calcium, potassium, mainly sodium, glucose, amino acids, they are all reabsorbed by the convoluted tubules. Only the nitrogenous waste, urea and uric acid, they are only sent out as urine. That is as a nitrogenous waste. So that is urine formation, mechanism of urine formation. What happens? The glomerulus, when it is eliminating the nitrogenous waste, it absorbs all the urine. It is eliminating the urine, and that is sent out through the proximal convoluted tubule, Henry's loop and distal convoluted tubule. And during this passage, sodium, potassium, calcium, glucose, amino acids and other useful substances, they are reabsorbed in the convoluted tubule. Only urea, uric acid in the form of urine is sent out, and that is collected, that is reaching the collecting duct, and from the collecting duct in the medulla portion it is emerging out as ureter, that is temporarily stored in the urinary bladder. Urine is temporarily stored in the urinary bladder, and when it is full, it is sent out through urethra. This is formation and disposal of urine, mechanism of urine formation. I hope you understand this. So this is part of transportation where the excretory products are transported. Are you clear? So this is for our lesson transportation in animals. Okay. So we will continue in the next video session transportation in plants. I hope you understand. We will continue in the next session. Thank you.